Good morning, good afternoon, and good evening, everyone, and welcome to the 7th webinar of Obesity 2020, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. We've lined up a number of webinars, all focused on the science and technology advancing obesity research. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This webinar is titled Animal Model Selection, Study Design, and Current Trends in Preclinical Obesity Research, and will feature Dr. Fred Beasley, Director of Scientific Engagement, Cardiovascular and Metabolic Disease at Crown Bio. Dr. Beasley will give an overview of study design, criteria for defining your study aim, and expert advice for selecting the right rodent model for your preclinical obesity research. This webinar has been sponsored by Crown Bio, so a big thanks to them for making this event possible. I'm very pleased to welcome Fred Beasley. Fred, thanks so much for joining us today, and feel free to take it away.
Thanks a lot, Liam. That was a great introduction. Thanks for joining us today, everybody. Today's webinar is going to serve as a broad level overview of factors you should consider when you're trying to pick an appropriate preclinical rodent model for studying obesity and its treatments. I'm going to structure the presentation as follows. I'll present an overview of trends in obesity and associated illness and the role of pharmacological intervention. I'll discuss figuring out what your study's aims should be. I'll introduce you to commonly used rodent models, including induction of obesity through dietary means, as well as obesity resulting from genetic predispositions. And I'll present additional considerations you may have for study design. Obesity is a medical condition where body fat accumulates to a point where negative health effects occur. This really just represents an imbalance favoring caloric intake over caloric expenditure. This can happen as a combination of either and or excessive food consumption and insufficient physical activity. Genetic factors can also drive the magnitude of either of these contributors. Obesity is graded on the BMI scale, which is calculated as one's body mass divided by the square of one's height. A BMI between 25 and 30 is considered overweight, and BMIs exceeding 30 fall into different categories of obesity severity. Excess adipose tissue is chronically pro-inflammatory, which negatively impacts insulin sensitivity both within that adipose tissue and systemically around the body. Adipose tissue secretes numerous bioactive models like pro-inflammatory tumor necrosis factor and interleukin-6. Excessive liver fat can lead to oxidative damage within this organ. These factors have detrimental impact on the insulin sensitivity of key glucose-consuming tissues, including muscle and liver. This explains why people who are obese are approximately 10 times more likely to develop type 2 diabetes, contributing to the term diabesity. Risks escalate sharply from a BMI of approximately 30. Excess adiposity is a well-characterized driver of disease states arising from chronic inflammation and insulin resistance, including hypertension and liver disease. An American meta-analysis of indication-specific healthcare costs in the obese population relative to the non-obese population uncovers that extra spending is required for a wide variety of acute and chronic illnesses in the obese population, including heart disease, stroke, chronic airway obstruction, fatty liver, sleep apnea, and depression. The recent COVID-19 pandemic has exposed another consequence of the obesity crisis. Multiple meta-analyses have been conducted during the first 6 months of the pandemic, and they confirm that obesity is a strong, independent risk factor of more severe outcomes from infection, including admission into intensive care and acute respiratory distress syndrome. Popkin et al. Performed the most comprehensive analysis that I've read during the early wave of infection, and they determined that obese individuals have a 46% higher risk rate to test positive for COVID-19, a 113% higher risk rate for hospitalization, a 74% higher risk rate for admission to intensive care, and a 48% higher risk rate for death. Other studies have shown that each increase in BMI obesity category is associated with a 10 to 15% increase in the likelihood of a hospitalized COVID patient requiring mechanical ventilation, and severely obese patients are over 85% likely to require a ventilator. Severely obese patients with a BMI over 35 have drastically increased likelihoods for respiratory and are more than 12-fold at risk for mortality. Global obesity rates have tripled since 1975. Currently, about 13% of the global adult population and 40% of the American adult population are obese. Diet and physical activity should usually be a first-line treatment prescribed to combat overweightness. However, studies have shown that behavioral changes can be difficult to maintain because they induce lasting hormonal changes that are conducive to weight regain. A seminal study by Sumethran et al. Reveals that patients on a weight loss plan sustain persistent changes to the secretion patterns of their brain-gut axis hormones, even over a year after starting their plan. These include an increase in postprandial production of ghrelin, the hunger hormone, and decreases to the anorexigenic hormones peptide YY, amylin, and cholecystokinin. This phenomenon highlights why changes in diet and exercise may be insufficient for many obese patients and why pharmacotherapeutic intervention can be valuable. The NIH recommends pharmacotherapy for patients with a BMI over 30 or for patients with a BMI of 27 to 30 who suffer at least one comorbidity. The FDA has currently approved 4 medications for long-term weight loss. A 5th, plorcaserin, was recently voluntarily withdrawn from American markets by Eisai Incorporated after a post-marketing clinical trial determined a modest but significant increase in cancer rates in the lorcaserin treatment groups. The FDA approves 3 more drugs for short-term weight loss. These act through a variety of mechanisms, including inhibition of fat uptake as stimulants and as antidepressants. Liraglutide is the one prescribed anti-diabetic that's also approved for weight loss. It's a GLP-1 receptor antagonist that improves glucose metabolism and diminishes appetite. However, other anti-diabetic medications are often prescribed off-label for obesity, such as the sodium glucose transporters like canagliflozin, insulin sensitizers like metformin, and the amylin analog pramlintide. Regardless of on or off-label prescription status, weight loss drugs should be used as adjuncts to and not surrogates for modifications to dietary choices and physical activity. Those should always be the first-line treatment. I'll now commence a review of commercially available rodent models that are commonly used for modeling obesity and for developing preclinical obesity drugs. Rodents can be great subjects for a variety of reasons. These include strong conservation with humans for hormones, receptors, and enzymes that are relevant to metabolism. Rodents are predisposed to developing obesity as a consequence of confinement, and they can develop disease in a relatively rapid time frame. They have genetic tractability, especially mice, so they can be modified for therapeutic or translational improvements. They're conveniently sized for economy of drug manufacturing, and they've become abundant and affordable commercial commodities that can be available at most local scales. However, no model is perfect, so you need to acknowledge that each one's going to have limitations that are inherent to it. A key one that's similar to most rodent models is that they're housed in small enclosures where their physical activity is limited, and they're given free, unlimited access to food. So many researchers don't have the luxury of choosing a diet that is not specifically defined by the researcher. There's no food choice options unless models are sophisticatedly elaborated. It's important to define and clearly define a focus so you can accentuate obesity in ways that enable treating a target of interest. Are you simply looking at altering appetite or energy consumption, or are your readouts crucial to measures of metabolic disease like hyperglycemia and insulin resistance, or to comorbidities like liver and kidney disease? Despite their limitations, a measurable phenotype or pathology with a quantifiable window for therapeutic efficacy is typically sufficient, even when animal obesity does not mimic all facets of the human condition. I'll walk you down 2 avenues of selecting the appropriate model. The first deals with diet-induced obesity, which is often referred to as DIO. This can involve administration of a straightforward high-fat diet. High-fat diets can further be enhanced with adjuncts that are overrepresented in modern human diets to induce comorbidities. The second avenue deals with obesity that arises from genetic variations that predispose animals towards obesity. These can be monogenic, i.e. a gene mutation or an allele that affects a single pathway, typically leading to compromised satiety and overeating. Or these can be polygenic, where multiple genetic factors, often uncharacterized, contribute to alterations in food consumption and or metabolism, generating an animal with accentuated predisposition to obesity. Polygenic animals may develop obesity on normal chow diets, but are often accelerated to obesity with high-fat diets, and can be accelerated to comorbidities with high-fat diets containing adjuncts. Let's get started with rodent models for diet-induced obesity. High-fat diets are modeled after the modern Western tendency to consume a relatively higher percentage of total kilocalories derived from fat than was historically typical, or than is observed in developing nations. The global average of dietary energy consumption provides a total of 17.4% of energy in the form of fats, but the average in high socioeconomic index countries provides a total of 26% energy in the form of fats, in addition to a higher daily amount of total calories. The American diet is among the worst of those high socioeconomic index countries. It provides between 30 to 40% of total energy in the form of fats. Fundamentally, these models involve transitioning animals from a low energy density diet, one low in fat and high in complex carbohydrates and fiber, to a high energy density diet, one that's high in fat and low in carbohydrates. Rats and mice are both responsive to this adjustment with differing degrees of uniformity. Reports can vary, but for rats put on a high-fat diet, including Wistar, Long-Evans, and Sprague-Dawley, the emergence of a diet-induced obesity phenotype is typically observed among 40 to 60% of individuals of both sexes, suggesting there is some polygenic underlying basis for the DIO phenotype. Mice have more consistent emergence of the DIO phenotype within strains, but to different degrees between strains. The C57 Black 6 mouse, notably the substrains 6J, maintained by Jackson Laboratory, 6N maintained by Taconic Biosciences are popular choices. They exhibit a high rate of very strong DIO phenotypes, up to 90% in males. It can't be worthwhile to screen subjects in the early weeks of a diet to weed out DIO individuals from DIO non-responders. The male C56 Black 6J mouse is considered a gold standard test animal for high-fat diet studies. In my experience, it has dominated the literature for obesity studies using wild-type mice. The Sprague-Dawley rat is also widely used. These animals exhibit numerous features of metabolic syndrome and obesity, making them useful for targeting appetite, weight loss, insulin sensitization, glycemic normalization, and cholesterol control. Rats exhibit some characteristics of fatty liver-like disease, but less so than mice. More so than mice. Control selection is also important. You should decide if animals on a low-fat diet are appropriate. Alternately, cohort mates resistant to presenting obesity may provide valuable genetic or biochemical cues into the mechanism of action for novel therapeutics. A key limitation of traditional high-fat diet models is they do not generate many of the advanced comorbidities that develop among obese patients. High-fat-fed rodents exhibit features of compensatory insulin production characteristic of the pre-diabetic state, but they don't exhibit pancreatic beta cell pathologies, so are not really suitable for studies on frank type 2 diabetes. They will also not develop advanced non-alcoholic steatohepatitis or NASH without further dietary adjuncts. Before committing to a DIO study, you should perform a review of the literature to be aware of variability-inducing factors that can affect outcomes. First and foremost, there's a significant variety in the composition of diets that have been reported in the literature. This includes the percentage of calories that are derived from fat, the origin of the fat, including animal and vegetable sources, and the micronutrient and protein content of the feed. These can affect all parameters of your study, including weight gain, comorbidity phenotypes, and the length of the study required to achieve endpoints. You should always try to use a highly defined, standardized research diet and have discussions with your feed supplier on how to minimize variables that aren't related to fat energy content. Additionally, reports range widely on the responsiveness of rodent substrains to the obesogenic diet. Male Black 6 mice of the J substrain are reported to convert at very high rates, while colonies in the N substrain are strongly resistant. This may be due to a mutation in the J substrain's nicotinamide nucleotide transhydrogenase gene, which is involved in beta cell mitochondrial metabolism. However, the taconic-specific sublineage of the N substrain has an intact NNT and doesn't present this variability, suggesting contributions from other gene loci. Wistar and Sprague-Dawley rats are well known to respond variably to DIO feeding, with some studies reporting only a 50% prevalence of the obesity phenotype. Conversion rates may also be attributable to the breeding facility from which the animals are sourced. This means it is very important to screen out DIO responders during early weeks of dietary intervention to ensure you can enroll suitably obese-prone subjects. This may add significant cost to a project, but it can also provide you cohorts of lean animals to serve as controls. Obesity studies can be very time-consuming, potentially running in excess of 20 weeks if you want to achieve morbid obesity. If you know your therapeutic window of time and you want to bypass costly in-house induction time, a lot of suppliers are now providing off-the-shelf animals that have been pre-induced to the appropriate stage of obesity. This can also eliminate the need to order surplus animals so you can screen out DIO from DIO non-responsive animals. Be aware that transit-associated stress can result in a temporary halt or even a reversal of weight gain, so you have to factor in appropriate reacclimation time when you purchase off-the-shelf obese animals. Finally, a high-fat diet is usually insufficient to generate salient comorbidities, including NASH and frank diabetes. So next I'll discuss additional dietary modifications that you can impose to improve the development of these comorbidities. Human obesity is associated with an increased risk of non-alcoholic fatty liver disease and non-alcoholic steatohepatitis, or NAFLD, and NASH. There are no approved drugs for NASH despite widespread prevalence of this disease. Inclusion of other harmful components of the Western diet have led to mouse models dietary mouse models for NAFLD and NASH. Increased consumption of fructose, typically as sucrose or corn syrup, is a key driver of the human obesity crisis. Fructose fails to induce insulin and leptin secretion and does not cause satiety. Furthermore, it's been shown to cause increased leptin resistance in rats when consumed chronically, although a similar effect in mice is debated. Nevertheless, fructose is estimated to contribute about 9% of the average daily energy consumption in the American diet, and this is a relatively recent introduction on the evolutionary scale. Trans fats, saturated fats, and even long-chain mono or polyunsaturated fats have also been shown to have some deleterious effects on metabolism and inflammation in the liver. Numerous mechanisms have been proposed and are still subject to conflicting reports, but diets enriched in these types of fats seem to increase production of atherogenic, low-density lipoprotein, increase hepatic inflammation through inefficient beta oxidation and augmented lipotoxicity, and to increase insulin resistance in hepatic cells. In a recent study by Liu et al., C57 black 6 mice on a high-fat diet with fructose and glucose in drinking water were shown to accelerate towards NAFLD and NASH. Fructose and glucose alone did not contribute to total body weight, but they did increase risk-associated visceral fat when incorporated into a high-fat chop. They also increased liver triglycerides and free fatty acids and increased circulating liver and cholesterol levels. Including these adjuncts led to worsening of the parameter components of the NAFLD activity score over feeding a high-fat diet alone. These scores include hepatocyte ballooning, inflammation, and steatosis. Fructose and glucose also strongly enhance the amount of fibrosis occurring in the liver during a high-fat regimen. They also increase liver dysfunction as measured through liver-generated transaminase enzymes as serum markers. These routine endpoints for NASH quantification were evident in as little as 8 weeks of dietary induction, highlighting the dangers associated with a fast food model diet. The severity of fibrotic NASH may also be improved by substituting the types of oils milled into high-fat diet chow. Typical rodent feeds incorporate a high degree of unsaturated, vegetable-derived oils as part of their fat content to a total fat composition of 10 to 20% in terms of caloric content. In contrast, the GAN and amylin diets, AMLN, are 40%, and I apologize for the typo on the screen, should be 40% fat by caloric content, where a significant portion of the fat content is derived from saturated fat or a combination of saturated and trans fats. The GAN diet is more frequently published recently as the recent FDA ban on trans fat has limited the production of the AMLN diet. GAN and AMLN also contain high amounts of fructose, sucrose, and cholesterol. They present a very severe combination of the worst attributes of the Western diet. In the C57 black 6 mouse, these diets can be used to reliably generate obesity with some insulin resistance and metabolic syndrome, although crucially they lack overt hyperglycemia. Obesity is accompanied by robust increases in the parameters of the NAS score, especially steatosis and inflammation, and increases in fibrosis indicative of NASH. Halting or reversing fibrosis is a crucial primary endpoint and a notoriously difficult one to achieve for NASH drugs being evaluated in advanced clinical trials. Resolution of steatosis is the other key primary endpoint for these trials. The GAN diet provides robust degrees of both these disease hallmarks, but fibrosis requires a substantial amount of time before reaching satisfactory severity, with onset occurring around 27 weeks of diet. However, it progressively accumulates from there. A researcher may not have time to wait for this development, but pre-induced animals are now becoming commercially available from major suppliers of lab animals as an off-the-shelf commodity. These should rapidly accelerate preclinical development programs and accelerate anti-NASH pipelines towards the clinic. I'll now proceed to discuss rodent models of obesity where there is an underlying genetic basis for disease development. As I've discussed, diet-induced obesity models have numerous useful applications, including for studying weight gain, adiposity, and many of the metabolic dysfunctions associated with the pre-diabetic state. They're also useful for studying the comorbidity of hepatic steatosis, and with nutrient modifications, they can be used to generate NAFLD and NASH. But they don't exhibit hallmarks of phase 1 to phase 3 diabetes, including beta cell hypertrophy and atrophy. For studies on overt diabetes, we should turn to models where the basis for disease is genetic. I'll start by looking at the monogenic leptin mutants, which have paved the way for numerous discoveries in the field of metabolic disease. In the 1960s, mouse breeders at the Jackson Laboratory isolated 2 spontaneous mutant types that overeat and become obese and diabetic. One was termed OB for its more severe obesity phenotype. The second was termed DB for its more severe diabetic phenotype. Studies revealed a circulating factor from the DB stock could rescue the food intake and energy metabolism defects of the OB stock, but not vice versa. These studies paved the way for discovery of the satiety hormone leptin. This is a key player along the brain-adipose axis. This peptide is secreted by white adipose tissue proportionally to stored triglyceride levels. It targets the hypothalamus, promoting satiety and stimulating metabolic rate. The OB knockout ablates leptin synthesis, and the DB knockout inactivates the leptin receptor. Obesity results as a combination of excessive eating and reduced energetic expenditure, including reduced thermogenesis. Leptin knockout mice are overweight even when restricted to a normal caloric diet regimen. Obesity is apparent as early as 4 weeks of age, and this obesity rapidly increases. In the black 6 background, hyperglycemia increases until 8 to 12 weeks of age, plateaus, and then is compensated for by pancreatic hyperplasia. Mice demonstrate glucose intolerance, however, this is maintained throughout their life. In contrast, in the BKS background, hyperglycemia rapidly rises to over 600 milligrams per deciliter and is not compensated. This leads to pancreatic beta cell atrophy and severe depletion of pancreatic cell islets, with death occurring by 10 months of age. Leptin resistance is a common phenomenon arising during human obesity, but leptin deficiency is only very rarely a cause of obesity in humans. This calls into question the translational value of these models. Researchers frequently use the OB-OB mouse to evaluate anti-obesity medications that target appetite suppression and caloric expenditure. They're also useful to evaluate leptin as a co-treatment for additional therapeutic benefits. The DB-DB mouse in the BKS background is used to evaluate the efficacy of anti-diabetic drugs, including interventions that may benefit stage 3. It's also useful for therapies targeting hyperlipidemia. It's important to comprehensively study the total set of phenotypes presented by your strain to make sure you choose a lineage that best fits your research needs. Leptin mutations also exist in rats. The Zucker fatty rat mutant was discovered in an outbred colony in the 1960s. The trait demonstrates Mendelian inheritance for a recessive allele, leading to profound obesity and elevated serum lipoprotein. These rats exhibit insulin resistance and impaired glucose tolerance, but are able to maintain normal basal glucose levels through increased insulin secretion. The mutation was eventually mapped to the leptin receptor gene, which is analogous to the mouse DB allele. Selective inbreeding of Zucker fatty rats for hyperglycemia generated a lineage called the ZDF rat, where males consistently develop spontaneous hyperglycemia, and females can be reliably induced on a high-fat diet. The diabetic phenotype is attributed to an autosomally inheritable genetic defect in beta cell transcription that segregates independently of the FA locus, and is silent in the absence of obesity-induced insulin resistance. The Agouti yellow mouse has also been another popular strain for obesity research studies. The Agouti peptide is normally expressed only by melanocytes in the adult mouse, and it induces a golden pigmentation phenotype in fur. In obese mutants, however, a genomic deletion has fused the Agouti gene to a constitutive pleiotropic promoter, leading to its expression all over the body. When ectopically expressed, the Agouti peptide serves as a powerful antagonist for melanocortin receptors 3 and 4 in the hypothalamus. These receptors are important for regulating pathways that modulate feeding behaviors, satiety, and energy expenditure through mechanisms like thermogenesis. The MCR4 receptor is associated with between 1 and 2.5% of human obesity, so this model may have more translational benefit than a leptin signaling knockout mutant. Agouti also potentiates insulin's ability to promote lipogenesis and to inhibit lipolysis in adipose tissues. This leads to adipose proliferation and hypertrophy. This table summarizes monogenic mice at a glance. The OB and DBD mice in the black 6 background are monogenic mutants for leptin signaling. Suitable controls include wild type or heterozygous animals. In their case, obesity is severe, achieving over 40% of body weight. Their basal hyperglycemia is moderate and remissive with aging, but their insulin resistance is severe and rapid. They are hyperinsulinemic, hypercholesterolemic, and they exhibit mild hyperlipidemia. They do exhibit beta cell changes, including hyperplasia and hypertrophy, and they're useful for obesity studies, for metabolic syndrome, type 2 diabetes, and steatosis. The DBDB mutant in the BKS background is also a monogenic leptin receptor mutant. Suitable controls include heterozygotes or wild types. It exhibits moderate obesity, but basal hyperglycemia is severe and sustained. Insulin resistance is severe and rapid, and hyperinsulinemia is soon decompensated, becoming hypoinsulinemia, making these good models for studying frank diabetes. They're also hypercholesterolemic and hyperlipidemic. They're useful for studies on obesity, metabolic syndrome, type 1 and type 2 diabetes, and fatty liver disease. The Agouti mouse is a monogenic mutant for overexpression of the Agouti peptide. Controls include wild type or non-Agouti animals. These exhibit high degrees of obesity and moderate hyperglycemia progressing to severe. Insulin resistance is severe and rapid, and later becomes hypoinsulinemia, uncovering that they also decompensate. They're hypercholesterolemic and severely hyperlipidemic. They exhibit beta cell changes and are useful for studies in obesity, metabolic syndrome, type 1 and type 2 diabetes, and steatosis. Zucker fatty rats are also monogenic leptin receptor mutants. They have controls including wild type or heterozygous animals. They exhibit severe obesity but no basal hyperglycemia, although they are insulin resistant and hyperinsulinemic and hypercholesterolemic and hyperlipidemic. They do exhibit some beta cell changes and some fatty liver disease. They can be induced towards NASH when put on a high-fat diet. They're useful for studying obesity, metabolic syndrome, steatosis, and type 2 diabetes. The Zucker diabetic fatty rat is also a monogenic mutant receptor predisposed to beta cell transcription defects. Controls include the Zucker wild type or heterozygous animal. They exhibit mild obesity and severe basal hyperglycemia, which rapidly progresses to severe insulin resistance, leading to diabetes and actually very rapid onset of death. They exhibit early failure of beta cell changes, do not develop fatty liver disease. They're useful for type 1 and type 2 diabetes, as well as studies on advanced diabetic comorbidities. When considering the value of a monogenic mutant for obesity in rodents, don't be overconfident in the translatability. Fundamentally, the genetic underpinnings of human obesity and associated conditions are polygenic. They're rarely due to disturbance of a single factor. Only severely rare cases of obesity result from inactivation of leptin signaling. Leptin signaling mutations in rodents are also abnormally strong phenotypes. Antagonism of melanocortin receptor pathways in the Agouti mouse is somewhat more representative of some human causes of obesity, but they also demonstrate an exaggerated phenotype in the mouse. Other single gene inactivations have been uncovered, but their phenotypes are very cryptic. For instance, the GLP-1 receptor knockout mouse is glucose intolerant, but doesn't exhibit increased food uptake or obesity. Molecular biological approaches to knockout genes of interest is a powerful way to confirm mechanisms of action of drug candidate, but these mutant strains have very little predictive value. Most obesity-related genes are involved with neuronal processes with a large degree of inter-individual heterogeneity, and potential codependence on other alleles to present a phenotype. Animal strains that present a polygenic origin of obesity are important for their ability to better model the human condition. Among polygenic mouse strains, the most recent entrance to the commercial market is the MS-NASH mouse. This was formerly called the Fatso mouse. MS-NASH stands for Metabolic Syndrome and Non-Alcoholic Hepatic Steatosis. It was generated by crossing obesity-prone C57 Black 6J mice and AKRJ strains with selective inbreeding for obesity, insulin resistance, and hyperglycemia phenotypes. The functionality of the leptin pathway is maintained in the MS-NASH mouse. Nevertheless, it develops spontaneous obesity even on a low-fat diet, comparable to Black 6 mice on a high-fat diet. The MS-NASH mouse is hyperglycemic, hyperinsulinemic, hypertriglyceridemic on a standard diet, and shows impaired glucose tolerance. Standard of care diabetic therapeutics like the GLP-1 receptor agonist semaglutide are effective at managing blood glucose, demonstrating that this is a translational model for metabolic syndrome. The MS-NASH mouse can be fed a westernized diet, including 40% fat, mostly from butter, as well as 0.15% cholesterol and fructose in drinking water. This westernization enhances its weight gain and adiposity and makes it hypercholesterolemic. The westernized diet treatment results in hepatomegaly due to accumulation of hepatic triglycerides, indicative of NAFLD. Histology confirms early stages of steatotic liver disease. The parameters of the NAS score, including steatosis, hepatocyte ballooning, and lobular inflammation, are significantly accentuated by feeding a Western diet within as little as 16 weeks. The MS-NASH mouse also begins to exhibit signs of fibrosis as early as 16 weeks on diet. This is accompanied by increased amounts of non-invasive serum markers of liver disease. Among polygenic rat strains, the most recent entrance to the commercial market is the ZDSD rat. This was generated by crossing the lean, leptin-intact ZDF diabetic rat with an inbred, high-fat diet obesity-responsive substrain of the Sprague Dawley rat. The cross was selectively inbred for obesity, insulin resistance, and hyperglycemia phenotypes. It develops very early onset obesity compared to the outbred Sprague Dawley rats as controls. Additionally, the ZDSD rat has increased abdominal fat and dyslipidemia. The ZDSD rat is a valuable model for studying type 2 diabetes with similar development to what is seen in human age-related onset of diabetes. This includes a period of hyperglycemia paired with compensatory hyperinsulinemia. The prediabetic state lasts from 8 to 16 weeks of age. Then there's a period of insulin resistance followed by insulin decline, resembling the overt diabetic state. Finally, there is beta cell failure accompanied by overt complications, including fatty liver and hypertension. The ZDSD rat also exhibits key features of diabetic nephropathy. These include progressive albuminuria and an increase in the urinary kidney injury marker, TIM1. There's progressive enlargement of the kidneys and progressive thickening of the kidney basal membrane. The ZDSD rat is responsive to standard of care drugs like lisinopril, demonstrating that this model of obesity-related diabetic nephropathy has translational value for preclinical drug development. To conclude the webinar, I'd like to introduce you to some study design considerations that are typically overlooked but should not be disregarded. Many rodent obesity models rely on overconsumption of calories, including high-fat diets and leptin knockout phenotypes. Models are less frequently designed to consider mechanisms related to caloric expenditure. This means they often disregard endpoints designed around physical activity, thermogenesis, food choice, and other feeding behaviors. These can be evaluated using automatable electronic monitoring and feeding systems, which are designed to provide additional stimuli and data while reducing technician burden and animal disruption. Pictured here is the Comprehensive Lab Animal Monitoring System, or CLAMS. Automating food delivery can stagger meals into predetermined time slots to compare food intake ad libitum relative to at times more attuned to an animal's circadian rhythms. This can be used to dispense normalized amounts of calories or nutrients between obese and control groups to determine if obesity is a condition of altered metabolism rather than one of increased consumption. Monitored running wheels can track voluntary engagement in physical activity to quantify an animal's willingness to exercise. Alternative mechanisms can be used to track an animal's movements in the X, Y, and Z planes for more comprehensive analysis of total physical activity. Implanted telemetry devices can report real-time data on body temperature and glucose. This can be used to assess an animal's level of thermogenesis, which is the burning of brown adipose tissue to produce heat. Gas sensors can measure the flow rates for oxygen consumption and carbon dioxide emission. These can be used to indirectly calculate calorimetry as a function of time. Another interesting consideration is that very few pharmacological compounds have been tested in rodents at thermoneutrality. This is the ambient temperature at which a mammal's thermoregulatory mechanisms are inactive. More casually put, this is the temperature at which an animal feels comfortable. Most labs subject rodents to ambient temperature that is thermoneutral for humans in the range of 22 to 26 degrees Celsius. In reality, for mice, the thermoneutral zone is 30 to 32 degrees Celsius. This means that at human ambient temperature, they're constantly engaging in adjustments to maintain a comfortable state. They may expend up to a third of their energy to maintain a constant body temperature. This is achieved through two principal mechanisms. First, the animal engages in more physical activity. This is accompanied by increasing food intake, but to a lesser degree than is required to compensate for the increase in exercise. More importantly, the animal activates thermogenesis within brown adipose tissue. Under conditions of cold stress, the sympathetic nervous system will release norepinephrine to activate brown adipose cells. This stimulates oxidation of fatty acids for mitochondrial respiration. Free fatty acids de-repress mitochondrial thermogenin, also known as uncoupling protein 1. This uncouples electron transport and ATP generation, causing respiratory energy to be released as heat instead. Studying rodents at their own thermoneutral temperatures, where the sympathetic activation is not chronically in play, can result in enhanced obesity phenotypes and alter the magnitude of comorbidities. The animal is affected systemically, showing decreases to blood pressure and heart rate, diminished production of endocrine, catecholamine, and corticosteroid hormones, and decreased thermogenic energy expenditure. Studies have also shown that animals on high-fat diets develop more severe liver diseases when fed high-fat diets at thermoneutrality. Parameters including liver weight, triglyceride content, and inflammatory cell infiltration contribute to augmented NAFLD activity scores and higher levels of markers of liver disease. This can create larger windows for therapeutic efficacy or alter the efficacy of the drug itself. A proof of concept study by Shao et al. Reveals how preclinical candidates can be more effective at thermoneutrality. The pharmacological benefits of a beta-adrenergic receptor agonist, which increases fat tissue lipolysis and brown adipose thermogenesis, are only readily apparent when mice on a high-fat diet are housed within their thermoneutral zone. In this state, there are more profound effects on decreasing food intake, reducing body weight, and reducing the body's proportion of dangerous white adipose tissue. Future studies into drugs targeting pathways that are completely removed from uncoupled thermogenesis should illuminate if thermoneutrality can uncover benefits of other new or existing pharmaceutical agents in combating obesity. To summarize, high-fat diets provide a model, a relatively easy model for easy route to model obesity. They offer weight gain and adiposity, steatosis, and metabolic syndrome. Adjuncts like fructose and saturated and trans fats, as well as cholesterol, can adapt models for NASH. These also provide a more translational percentage of calories from fat and generate some characteristics of metabolic syndrome, although crucially, they usually lack hyperglycemia. Leptin and MCR signaling mutants can offer a rapid path to obesity and type 2 diabetes, but may lack translatability. Polygenic mutants, on the other hand, have many modalities for studying obesity, diabetes, and NASH, and are potentially more translationally relevant. For other considerations, technological and environmental upgrades to husbandry and monitoring will add translatability to studies on energy expenditure. Thanks everyone for tuning into our webinar. If you'd like to get in touch with us about discussing obesity and metabolic disease models, please email bizdev@crownbio.com to contact your local account manager, or visit crownbio.com to learn more about our comprehensive translational metabolic disease platforms.
Excellent. Thanks so much, Fred, for the fantastic presentation. And we are going to move on to the Q&A. So let's jump right in here. The first question today comes from William, who has asked, Fred, whether you would consider the DIO model to be more physiologically relevant to human health.
Again, it's going to depend on what your readout is. The advantage of the DIO model is that it's very easy to achieve, and there are currently now highly standardized diets that will allow for easy reproduction in previous studies. It does not it omits several crucial factors of the Western human diet that contribute to comorbidities, but it does provide phenotypes, including insulin resistance, that if you're targeting those are sufficient. And it's also reversible, so you can evaluate the efficacy of drugs that will promote weight burning. Again, it really just depends on what your mechanism of interest is, but its advantage is definitely its simplicity and its broad popularity, allowing for more accurate comparison with historical studies.
Excellent. Great answer, Fred. Next question. Andre has asked whether age of rats or mice affect the severity of diet-induced obesity.
Yeah, it does. The older they get, the more resistant they become. So typically animals are induced at a very young age, usually around 5 or 6 weeks. That's the simple answer, yeah.
Excellent, thanks. Next question here from Inga, who's asked whether you could say something about the difference in the length and function of the colon and small intestine in rats versus mice, and whether you know if that has any effect on diet and obesity.
That is wow, that's a very nuanced question. I have not come across this topic in any of my consultations with clients. I do know that rodents have a very fast transit time in the matter of hours. I'm not sure how it compares to humans. But definitely, I mean, there's a lot of things you could look at there. I'm sure this is something I would really like to look into further. I'm sure it will definitely affect anti-obesity drugs that target nutrient absorption. So a drug like orlistat, which is a lipase inhibitor, I feel like that would definitely be relevant. But I'm not sure how you'd go about setting up a study other than surgically reducing colon length in order to quantify that. Something that's, yeah, I'll definitely have to look into that.
Yeah, definitely an interesting question. Next question here from Diana, who's asked whether the thermoneutral temperature is the same for all strains of mice.
Yeah, I have not. It's generally published as a range, typically 30 to 32 degrees Celsius. So I've seen a few reports where it goes as high as 36. It's a very recent topic to be considered, and so I don't know if anyone's done an empirical study to confirm that. But that would be very interesting and probably pretty easy to quantify just by looking at sympathetic activation and measuring levels of UCP1 protein. I think that would be a really good study to do for off-the-shelf suppliers to further characterize their strains.
Excellent. Next question here. What do you think would be the observation expected if a high-fat diet were to be replaced with normal chow?
Animals will typically revert or normalize their body weight, and there will be a normalization of some of the parameters like lipidemia and glucose levels. Excellent.
And I think in the interest of time, we'll make this next question the last one. It comes from Carolina, who's asked, well, there's a body of research suggesting that the refined low-fat diets complementary to D12492 and D12451 lead to rodents that are not actually healthy models. How would you feel about using standard chow as the control when the goal is to compare DIO to an ideal state of health?
Yeah, okay, I get it. I think that refers to the control versions of those two diets. So the ones that don't have that high fat content are not healthy models. I'd be interested in looking further into this. That's a new topic for me. Generally, again, it's going to depend on your specific readout, but it's currently widely acceptable to use standard chow as the control. I suppose we would have to look at how we're going to go about defining the ideal state of health in a mouse in the first place before we can specifically tailor a base and then a high-fat version of a diet to modulate between that healthy state and the disease state. But yeah, I'd be interested if that questioner can actually refer me to some of these bodies of research, because that would be something worth pursuing further.
Perfect. Well, Fred, thanks so much for all your fantastic insights today, both in your presentation as well as in the Q&A session here.
Thanks everyone for joining, and check us out at crownbio.com for if you have any further questions on our models.
Definitely, yeah. Big thanks to everyone for joining us today to attend the webinar. And last but not least, a big thank you to Crown Bio for sponsoring this event. And in closing, thank you again for taking part in this Inside Scientific webinar produced in partnership with the American Physiological Society. And we look forward to having you with us again soon. Have a wonderful day, everyone.